Hi everyone, welcome and thank you for joining me at the WCCI virtual conference. My name is Marinas Toman and today I'll be presenting my paper on a computational study of astroglial calcium homeostasis in a semi-isolated synaptic cleft. I am part of the Computational Neuroscience and Neural Engineering research team based at the Intelligent Systems Research Centre at Ulster University in Derry, Northern Ireland. My co-authors for this paper are Dr. John Wade, Professor Liam McDade, and Dr. Jim Harkin. So first I want to introduce some key concepts and experimental observations that led to this study, and also why computational modelling is used as a tool to study the brain and ultimately predict behaviours in the brain. So to begin, the brain cells that I am primarily studying are astrocytes, and these are a type of glial cell that perform many functions in the brain. They are known to enwrap many neuronal synapses in the hippocampus, which is an area of the brain believed to be important for memory formation. It is thought that astrocytes semi-isolate synapses to carry out many roles, but specifically they can control synaptic transmission through homeostasis of different ions like calcium, sodium and potassium. And spontaneous calcium microdomains have been observed experimentally in distal perisynaptic astrocyte processes, and as of yet there is no consensus on what may be responsible for or what the function of these microdomains are. And astrocyte processes are on a nanometer scale and are too small to perform experiments on, and this is the primary reason computational modelling is a useful tool to probe these areas of the brain. A recent model predicts the formation of calcium microdomains due to the reversal of the sodium-calcium exchanger, and in this work we build upon this model to show how these microdomains connect as a local supply of calcium to the synaptic cleft, which may be critical for synaptic transmission. So now we will look at the main aims of the study, and the graphic to your right just shows a schematic of how the brain cells, in particular astrocytes, this green looking cell here, interact with neurons, these blue looking cells here. And in particular, we're interested in this area here, so where the astrocyte enwraps the synapse between two neurons. The first main aim of this study is to capture and describe the astroglial pathway by which calcium microdomains can form in astrocytes, and to be able to deliver calcium to the synaptic cleft during neuronal excitability. Secondly, we want to test whether these microdomains are sufficient to supply enough calcium to the synaptic cleft, which can be critical for synaptic transmission and also synaptic plasticity. And finally, another main aim of this work is to lay the foundation for future work on how astrocytes can affect and control synaptic transmission and synaptic plasticity. Next, we will look at the methods that are used in this study. It's computational modeling study, so we use a first order mathematical framework to model the entire system, and this consists of conductance-based differential equations that model the system over time, and also it's a multi-compartmental model, so it has multiple compartments describing different cells and compartments of the brain. Some of the channels that are used include the sodium-calcium exchanger, the NCX, the plasma membrane calcium ATPase, the PMCA, the excitatory amino acid transporter, or the EAT, the sodium-potassium ATPase, or the NKA, and voltage-gated calcium channels. This is a schematic of the entire study. So what we have here is, this is the perisynaptic astrocyte compartment, this green cell, And also we have a presynaptic neuron followed by a postsynaptic neuron. And we have this local extracellular space here, which represents the synaptic cleft. And we also have a global extracellular space, which represents everything outside these cells. And as you can see here, we have some membrane-bound transporters like the EAT, the PMCA, the NKA, the NCX, and the voltage-gated calcium channels, all embedded and exchanging ions between the astrocyte, the synaptic cleft, and the neurons. The compartments, as I say, include a presynaptic neuron, this perisynaptic astrocyte, semi-isolated synaptic cleft, or as we call it, the local extracellular space, and also a global extracellular space. Each compartment is filled with well-mixed concentrations of calcium, sodium, and potassium ions. So we will look at some results now from the simulations. And because this study is focused on calcium homeostasis, we will just look at the calcium currents from the astrocyte. So the simulations were ran stimulating the neuron from time of 10 seconds to 40 seconds. And the frequencies of the stimulation of the neuron was 10 Hertz, which is this first column, 20 Hertz, which is the second column, and 30 Hertz, which is the last column. Across the top, we have the calcium current from the PMCA. So as you can see, it's constantly extruding calcium from the astrocyte. But as the neuron is stimulated, this extrusion is increased. And this is a detail of 100 milliseconds towards the end of the stimulation. And on 20 Hertz, the calcium is extruded even further from the astrocyte. And as you can see here, it's the amount of extrusion is doubled. And similarly for 30 Hertz simulations, there's even more extruded, and in the detail you can see that that is triple the extrusion. So as the neuron is excited at higher frequencies, the calcium extrusion increases also. Now down on the second row here, we have the NCX current, and whenever the neuron is started to be stimulated, the NCX enters reverse mode and starts taking calcium into the astrocyte. And again, as you can see here in the detail in the inset, it's taking it in quite significantly. And also, as the frequency increases, the amount of calcium that's taken into the astrocyte also increases. The third row here is the calcium down along the process of the astrocyte, and this is quite a small current. So this is 10 to the minus 4 femtoamps. But also as the stimulation increases, the amount that is expelled from the astrocyte increases when it's going down the process. And then finally we have the calcium leak. So normally calcium is leaking into the cell, and as the neuron is stimulated more, the calcium is leaking in just that little bit more each time. So now we will look at the calcium concentration results. And the calcium concentrations are captured in 3 different compartments in the model, namely the perisynaptic astrocyte, the synaptic cleft, and the presynaptic neuron. As stimulation starts in the model, the NCX enters reverse mode, and calcium is taken in from the global extracellular space. And as you can see, the calcium concentration starts to rise in the astrocyte. At the same time, calcium is extruded from the astrocyte through the PMCA, and this begins the rise in concentration in the synaptic cleft. And during every spike of the neuron, calcium is taken into the presynaptic neuron at about 4 micromolar each time. As the stimulation frequency increases, we can see that this microdomain inside the astrocyte starts to rise a lot further, and it rises by about 10 nanomolar. And in the synaptic cleft, as the stimulation frequency increases, we can see that the synaptic cleft rises by about half a millimolar. And also in the presynaptic neuron, we can see the concentration rising to about 5 micromolar. And theoretically, this should be enough for synaptic transmission to occur. Now I will discuss the implications of these results and what we observed during the study. First, glutamate is released from the neuron during excitable periods, which causes the astrocytic EAT to be activated, and this leads to an influx of sodium into the astrocyte, which in turn leads to the reversal of the NCX. The NCX reversing causes a rise in calcium concentration in the astrocyte. The results show that this rise in concentration leads to a calcium microdomain forming, which then acts as a supply of calcium to the synaptic cleft. This shows astrocytes can influence synaptic transmission even in the absence of intracellular calcium stores like the endoplasmic reticulum. The results also demonstrate the concentrations of calcium, sodium and potassium are functionally coupled through transporters like the NCX, EAT and the NKA. For example, the EAT increases the sodium concentration, which then changes the behavior of the NCX, which influences the calcium concentration, and it also changes the behavior of the NKA, which influences the potassium concentration. Overall, these findings provide an alternate theory to the glial transmission theory by showing that astrocytes can influence synaptic transmission without releasing gliotransmitters or any other kind of chemical aside from ions. So now we will look at some planned future work that this study has led to. First, an extension of this study is currently ongoing, which consists of localizing the EAT, the NCX and the NKA in the membrane to show what effects this has on calcium shuttling from the cleft into the astrocyte and back out to the cleft again during rest periods. This ongoing work is informed by working closely with experimental neuroscientists and their recent observations that the EAT, the NCX and the NKA co-localize in the astrocyte membrane. This study also forms the basis for future work that investigates the extent at which this calcium supply to the cleft affects synaptic plasticity. And it is hoped that the future work will lead to a better understanding of the underlying processes involved in synaptic plasticity and overall learning in the brain. Future work will also incorporate knockout studies that will investigate how dysfunctional astrocytes or astrocytic transporters may affect neuronal excitability and therefore brain function. This is a list of references that were used in this presentation. For the full list of references, please refer to the paper. Thank you for your attention, and I'll be happy to take any questions you may have. Thank you.